Here are the module objectives. List the principal factors that can activate the coagulation cascade. Describe the activators for both the intrinsic and extrinsic pathways. And draw a diagram showing the sequence of factors in the intrinsic, extrinsic, and common coagulation pathways when the coagulation cascade is activated. So, let's get your brain in gear and briefly review primary hemostasis, which is the first response to vascular injury. So, looking at this diagram, first we have to have damage. The blood vessel is ruptured. The epithelial cells release their interior tissue factor, which causes constriction of the blood vessel, which causes platelets to adhere to the tear and form a platelet plug. This illustration shows the steps in the formation of a fibrin-stabilized clot. A and B illustrate primary hemostasis, whereas C, circled in red, is secondary hemostasis, which involves a series of enzymatic reactions to form fibrin, which stabilizes the platelet plug and stops bleeding. Let's pause for a minute so that you can get out your coagulation cascade diagram. Okay, everybody's ready. So here is the coagulation cascade. You'll notice that it's made up of three pathways: the intrinsic, extrinsic, and common pathways. The intrinsic pathway is activated by enzymes and protein cofactors found in plasma. The extrinsic pathway is activated by enzymes, protein cofactors found in the plasma, in addition to tissue factor. Tissue factor is an activator that is not normally found in the blood plasma. It is also known as factor 3. So tissue factor is released into plasma when there is a tear or damage to the vascular system. The intrinsic and the extrinsic pathways both converge into the common pathway at factor 10, and that's when we begin the formation of our fibrin. There are three groups of factors that we need to learn about as we begin to put the coagulation cascade together. So let's begin with contact factors. These factors are found circulating in the vasculature and are involved in activating the intrinsic pathway. I have listed them here along with some of the common names. These factors are moderately stable and are not consumed during the coagulation process. A second group, the prothrombin group, is dependent on vitamin K, which comes from diet or from intestinal bacteria. The factors involved in the prothrombin group are factors 2, 7, 9, and 10. These are stable factors and they also are not consumed in the coagulation process. However, they are inhibited by the medication warfarin. The next group of factors are the fibrinogen factors. These are factors 1, 5, 8, and 13. These are consumed during the coagulation process to form fibrin. Fibrinogen is produced by the liver and can be increased during pregnancy, inflammation, the use of oral contraceptive drugs. Fibrinogen is also known as factor 1. Here are a few other factors that are involved in the coagulation cascade. Calcium ions, which are also known as factor 4. These ions are used in both the intrinsic and extrinsic pathways. An example is factor 12A needs calcium ions to activate factor 9. Platelet factor is released from platelets and is found circulating in the blood in the vasculature due to injury or tear. So platelets are triggered to move towards a tear in the vascular, and in the process they release platelet factor. Procoagulation factors. These are actually the coagulation factors 1 through 13. They are not activated, so they're called procoagulation factors or zymogens. So you remember we've already talked about some of these factors in the previous slides. So let's see, what do you recall about these factors? They're non-activated. Each factor has one or more names. One more. That's it. They have been assigned Roman numerals 1 through 13. If they're activated coagulation factors, the Roman numerals will be followed by a small a. Good job on those 3 recall items. As discussed in the previous slide, once factors are activated, they are designated by a Roman numeral followed by a small a. Except in most of the common pathway factors. In the common pathway factors, as you can see in the diagram to the right, the synonyms are used and not the Roman numerals, as you can see here. There's a prothrombin activator, a prothrombin plus calcium to a thrombin, and then down to fibrinogen and fibrin. And so instead of using our Roman numerals, we use the synonyms for these factors. Here is a short video produced by Katie Bowes on the coagulation cascade and some of the lab tests used to detect coagulation problems. It is about 7 minutes and contains a wealth of information. So you will need to make sure you have your coagulation cascade diagram handy. You can add to it while you watch this video. You can either click on this arrow right here, or you can copy and paste this link in your browser. Remember, once the video has finished, to come back and complete this lecture. Okay, so let's break down the cascade into the 3 pathways. We'll begin with the intrinsic coagulation pathway. Contact factors that trigger the activation of factor 12 are collagen of subendothelial cells, platelet granular content or platelet factors, bacteria, and clay. If you remember from the video, clay is found in the blue top tubes, and we use this to test the intrinsic pathway. Once factor 12 is activated, a series of enzymatic reactions takes place through the intrinsic pathway, through the common pathway, to fibrin being formed. We'll continue talking about intrinsic pathway activators. So we have the Kinnin system, and it's made up of prekallikrein, which is sometimes indicated with a PK or called the Fletcher factor. We have the high molecular weight kinnin, or the Fitzgerald factor, and both of these enhance or amplify your other contact factors. An example would be that your factor 12A plus your high molecular weight kinin converts your PK to calligrin, and calligrin will amplify the activation of factor 12 to factor 12A, and so it gets in a loop where it will create more activated factor 12. Okay, let's activate these factors in the intrinsic and common pathway. This is taking place near the loose platelet plug that has formed at a tear in a vein or an artery capillary. So we have an activated 12A, and in the presence of your high molecular weight kinin, it acts to convert factor 11 to factor 11A. Factor 11A, in the presence of calcium ions and platelet factor, will convert factor 9 to factor 9A. So let's take a minute to talk about factor 8. Factor 8 is an acute phase reactant, and it is consumed during the cascade process. It can be activated by factor 2 from the common pathway, which is thrombin. In its inactive form, it is found in the plasma bound to von Willebrand factor. So factor 8 non-activated is a complex, and it's considered an anti-hemophilic factor. Factor 9A with calcium ions will activate factor 8 to split into factor 8A and von Willebrand's factor. Factor 8A and factor 9A will attach to the platelet surface, and along with platelet factor and calcium ions will activate factor 10, and this begins the common pathway where fibrin will be formed. All right, let's look at the extrinsic coagulation pathway. Make sure you've got your coagulation diagram handy. As you see, the extrinsic pathway does not have as many factors involved, and it also converges on the common pathway at factor 10. The extrinsic pathway is triggered by calcium ions and tissue factor. These convert factor 7 into factor 7A. Now, factor 7A is a very powerful enzyme that converts factor 10 to factor 10A in the presence of calcium ions. So it's a very short and sweet pathway. Okay, we have made it to the common pathway. As you can see in the diagram to the right, both the intrinsic and extrinsic pathways converge into the common pathway at the point where factor 10 is activated into 10A. The combination of factor 10A, factor 8A, calcium ions, and platelet factor converts prothrombin into thrombin. Thrombin cleaves fibrinogen into fibrin strands with the help of calcium ions. Fibrin strands then bind factor 13, activating it into factor 13A, which acts as a stabilizer and cross-links the fibrin strands over the platelet plug. All right, it's time for a break. Please go back and review Chapter 32 reading, the video by Katie Bowes, and work on completing your coagulation cascade diagram. Then You will want to copy and paste this link from Khan Academy for a more detailed explanation of the coagulation cascade. Next week, we'll start talking about testing, such as the PT and the APTT test.